All right, let us begin. We have a few material to get through. Before I forget, Monday. As much as we like seeing you guys, we don't want to see you on Monday. I keep telling my classes, I'm barbecuing and I'm napping on Monday. You should do something of such sorts. Okay. So, as a heads up, that puts Monday lab in a little bit of a pickle, actually. You guys are like, sweet, we have a day off. But that means you only get one day for histology. But the others labs get technically two. So it makes it a little bit more challenging, just heads up. So please keep up your studying. So that's that. Okay. Later tomorrow, you're going to have two things that are going to show up on Canvas. You're going to have another lecture quiz show up every week, lecture quiz, every week, lecture quiz. You're also going to have a lab quiz show up. So you have to do both lecture and lab. And as a reminder, lecture quizzes will be multiple choice. Lab quizzes will be like lab exams, so they are fill-in, and spelling will matter. Okay? Spelling will definitely matter. All right. It's open note. The quizzes are open note, open book, so you should be okay spelling it appropriately. And you can retake them until you get a perfect score on both lecture and lab, as many times as you like. They're really designed to help you prepare for the types of questions to expect on the lecture exam and lab exams, which are coming up towards September, mid-end September. Okay. Any questions before we begin today? Don't worry about putting this question on your card because we didn't really get to it last week. But I wanted to get your thinking about what can a cell do? What can a cell do? What can that cell do to affect diffusion? What is diffusion? What are you going to tell me? Movement of molecules, and I heard it many people say, from high areas of high concentration to areas of low concentration. So what can a cell do to affect, either to increase or decrease diffusion? Because cells actually do that. They change their properties to increase or decrease diffusion. Does anybody know what a cell can do to help that? Hopefully we can name at least one or two. Does anybody know what a cell will do to increase or decrease diffusion? Osmosis, to increase osmosis, which osmosis is another form of diffusion. It's just specific to water. Okay, what a cell is going to do to increase that is it's going to put proteins in here on the membrane, and those proteins will act as channels. So a cell can increase the number of proteins. More proteins, more stuff can go in. Less proteins, less stuff can go in. This is what your cells do so they can change diffusion. The other one that's really important and is really hard for us to visualize, what cells do is they will change the surface of their membranes. Some cells will make their membranes like this. They will make microvilli. By adding the number of microvilli, what does the cell do? It increases the surface area. And this is going to be a very important concept in physio. When you increase the surface area, you increase the area for diffusion. More area, more diffusion. You can increase the diffusion rate. Why do I mention this? Your gut cells, the cells in your gut, have a lot of villi, sorry. So remember, that's one way to identify simple columnar epithelial. It has a lot of microvilli. It doesn't have cilia. So it helps us identify cells as well. There are two types of cell division that we need to be familiar with in this class. For this first exam, I only care about this one. Mitosis is a form of cell division. This is when you get one adult cell and it's going to divide into two cells. And those two cells are called daughter cells. Daughter cells. So we'll make two daughter cells. Daughter cell 1, daughter cell 2. But mitosis makes two daughter cells that are identical. And that is important for you to recall. This feature for mitosis, they are identical. And what that does is that allows the body to maybe make one skin cell. And it's going to divide and become two skin cells. They are identical. When does the body do this? When does your body undergo mitosis? When does it do this, folks? For what purposes? Growth, maintenance, just to maintain things. Repair. You broke your bone. You need to grow new bone cells so they can repair themselves. Growth, maintenance, and repair. Your body will use one adult cell and they will divide into two identical daughter cells. This is different than meiosis. Meiosis is one adult cell is going to turn into not one, not two, not three, but four. 1, 2, 3, 4 sex cells. So this one's for reproduction. And what's very important is that those sex cells are not identical. They are unique. They are all very different from each other. This we will cover when we do reproduction, which is our last chapter. We're wrapping up our cell chapter, so we have to be familiar with mitosis. Mitosis. It starts with interphase. Interphase is kind of the growth and duplication phase. So this is when the cell is growing, it has just been born, and it's going to start some duplication so it can eventually divide and become two new cells. It starts with interphase. Interphase has three different stages. So interphase is broken up into three phases. Phase number 1 is just G1 phase. This is when the cell has just been born. And all it's doing, G stands for growth. And when it's growing, it's doing its normal functions. So muscle cells will do muscle cell things. Nervous cells will do nervous cell things. They will do their normal functions. And they start to grow. They have just finished dividing their brand new cells. They will grow and do their normal functions during G1 phase. The next phase is just called S phase. And S, I want you to remember this word, synthesis. Can anybody tell me what the definition of just the word synthesis means? What does the word synthesis mean? To make. And what this one is making during the S phase is this one is going to double the DNA. It's going to start to double things, particularly the DNA, not just the DNA, but other stuff. So when the cell divides, it's doubled it. So the two cells that come up at the end of mitosis now have one set. That happens during S phase, and S phase is over here. I forgot to show you G phase. G phase starts right here. So this is the start of life for that cell. So it'll go through G phase. It'll enter S phase. This is when it's duplicating. It's still doing its function, but now it's duplicating. So eventually it can divide and become two. Then it's going to enter its third phase. And this is another growth phase, but this is G2. And now it's growing. It's doing its functions, but it's going to get ready. It preps to divide. The end of G2 phase will officially enter mitosis. This is where mitosis occurs. This is the M phase for mitosis. And that is when cell division occurs. And cell division to make two identical cells. What I want you to remember for mitosis is this simple acronym. It sounds silly, which helps us remember it. IP MAT. You piss on the mat. That's how you remember it. IP MAT. What does I stand for? And remember, it has three parts. It has G1, S, and G2. What does P stand for? The first stage of mitosis, prophase. What does M stand for? Metaphase. What does A stand for? Anaphase. What does T stand for? Telophase. Those are the stages of mitosis, including the first part, interphase. Interphase technically isn't part of mitosis. So only the PMAT is the true mitosis phases. Here they are. We call them meiotic phases, the phases of mitosis. We're going to start by reminding ourselves IP MAT. Interphase isn't shown here. We already talked about G1, S, and G2 phase. We're going to talk about prophase. Prophase is over here. Prophase is the stage when the cell is going to prepare itself to undergo mitosis. So what is it doing to prepare itself? A few things start to happen. One, the DNA form into chromosomes. So they become concentrated. Eventually, they become these little X's. Let me zoom in on that real quick if I can. There we go. they become these X's inside the nucleus. That's a chromosome. So if you see those chromosomes form inside the nucleus, you are in prophase. It is preparing to divide. Prophase, to prep. The other thing that happens during prophase is the centrioles. They form their spindles. The centrioles form the spindles. And those are the little fibers that are coming off of them. That fiber, that fiber, that fiber, they started to form. So when you're in metaphase, look at how much longer those fibers are now. When they start to form, it is prophase. Those are the two steps I want you to remember. The DNA form chromosomes, the centrioles form for prophase. Metaphase. Metaphase. The word I want you to remember, middle. Why? Look at what the chromosomes are doing. Here they are. X, 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 X. They are lined up in the middle of the cell. The chromosomes line up in the middle of the cell. Oh. Why? So that they can decide which half goes to which side. This is bad dad humor. I say this is like when a divorced couple goes to the bank, puts all their assets in the middle of the table. You get this half, I get this half, because they're splitting up. Metaphase, the chromosomes are lining up in the middle. What does A stand for in IPMAT? Anaphase. The word I want you to remember for A is apart. Apart. The chromosomes get split down the middle and they go to each side. They are pulled apart. One goes to this side, one goes to this side. Half the money goes to dad, half the money goes to mom. Sounds terrible, but that's what's happening. The chromosomes are being pulled apart so they can go to either side. That is anaphase. What does T stand for? Telophase. I want you to remember 2. Why 2? You start to see 2 nuclei form. When you see 2 nuclei form, that means 2 cells are starting to form. You are in telophase. 2 nuclei, telophase. And at the end of this all, this pinches off. It's called cleavage. Cytokinesis for cleavage. And that is when the cells will cut themselves apart and form 2 identical cells. IPMAT. I need you to be able to identify the phase from an image or a description. What do I mean by that? That's from your book, by the way. Here we go. Interphase. This is when the cell is going through its G1, S, and growth phase. One thing about interphase is you only have 1 pair of centrioles. During prophase, the preparation phase, now you have 2 pairs of centrioles. And the spindles are formed. There they are, in between them. The spindles are formed. as are the chromosomes. That's an important one. Chromosomes. Metaphase, look at how nice they're lined up in both the histology and in the image. The chromosomes line up smack in the middle. Smack in the middle. What comes after metaphase? IPMAT. Anaphase. During anaphase, the key thing to remember is that they are being pulled apart. The chromosomes are being pulled to either side apart for anaphase. You can see them getting pulled apart. People are going to tell me, look, they're getting pulled apart in this image too. And I'm going to say, yes, you're right. Absolutely. But the key thing that's different between anaphase and telophase, so here's telophase, is the fact that now you can start to see two cells or two nuclei. Two. That's telophase. Eventually it will undergo cytokinesis, and cytokinesis is the, oh, that's supposed to be S, is the cut phase. It's cut into two cells. Okay? So from an image, I need you to tell me what phase it's in. From a descriptor, I need you to be able to tell me what phase it's in.
Would it be images like on the top or the bottom?
Either one. It could even be like this. On your cards, please. Check with your neighbor. Make sure you're good on this one. Before I move on, for lecture, for this chapter, we're going to focus on epithelial tissue and connective tissue. We're going to do muscle tissue when we cover muscles, nervous tissue when we cover nervous system. The reading guide will tell you what I want you to be familiar with in this chapter, or else you're going to be reading a lot more material than you need to know. Okay? So, tissues. We saw things like this. We saw histology. Anyone in here work with histology? No one does. Every now and then we get people who do histological staining. No one works with histology? Okay. Histology. Histology is the study of cells, particularly stained cells. That's why they look pink, purple, red. Sometimes they'll be black, blues, yellows even. The stains will stain parts of cells so we can visualize them. It's a hard topic. Many people are like, why are you making me do this? I don't understand. Why are you putting me through this? And let me tell you why. If I haven't told you, if I said this already, I apologize. You're going to have a patient that's going to come to you and it's going to be like, look at this mole. It looks a little weird. And be like, yeah, it does. You're going to have to take a sample of that mole. You're going to have to cut out a little square out of it. You're going to put it in a preservative. You're going to put it in a ball of wax. You're going to slice it super thin. Put it on a microscope slide. Now you have a thin slice of that mole, super thin slice of that mole on the microscope slide. What are you going to do? Then you're going to stain it. The typical stain, you're going to start hearing these, are E and H staining. This isn't important. Eosin and hematoxylin. What does that do? It stains the cells pink and purple. And right now you guys are like, that just looks like a bunch of pink and purple gobbledygook. Well, The dark, dense things that stain purple are dense bodies, like the nucleus. The endoplasmic reticulum will stain dark purple. The cytoplasm will stain light pink. Once you start to understand what the stain is doing, it is showing you the parts of the cell, the shapes of the cell, what the cells are made of. And that's what histology is for. So we can look at these cells, be able to identify them. That's what we're doing in class, just being able to identify them. But when you do this in the medical field, it's going to go beyond that. What you're going to find is you're going to be like, look, this cell right here, this one might be 500 micrometers in length. The normal is only 200. So you're going to have to measure these things or know how these things are measured. Why? When the cells are too big, what does that indicate? You're going to tell your patient, look at your skin. Look at this mole. Look at how much bigger it is. It has way more endoplasmic reticulum than it's supposed to. It's this type of cancer. Has way much more pigment than it's supposed to. It's melanoma. And based on its arrangement, it's stage 2 melanoma. We still use this to this day. It's an ancient art that is still used to this day. Okay. So we need to understand what tissues are and how they're arranged. Okay. I want to start this as a reminder that we need to understand what the parts of the cells do. Because tissues are made of 2 or more cells. So we need to understand what the parts of the cells do so we can identify what their roles are within a tissue. If I ask you this question, what is a tissue? What's the answer, folks? What are you going to tell me? It is a structure that is made of 2 or more cells that work together to complete a particular function, whatever it may be. Do we see cells here? Well, you betcha. There's one cell here, there's one cell here. This is one tissue. The darker stuff is another tissue. What are those 2 now? 2 or more tissues make a what? They'll make an organ. Okay. They will make an organ. So a tissue is 2 or more cells working together to complete a particular function. The cells have to connect themselves to one another. We talked about gap junctions. I need you to review those if you do not remember those. Tight junctions. What was the last one? The third one. Who remembers what the third type of junction was? It's not called a junction. That's the tricky part about that one. Desmosomes. That is what holds the cells together so they can form a tissue. The first type of tissue we're going to talk about is epithelial tissue. And epithelial tissues, it's important for us to recall that they line the surface of organs. And because they line an organ, what they do is they have a top side and a bottom side, but that's not the best word to use because sometimes they make little circles, sometimes they make tubes, but they still line the surface. An epithelial tissue will always have what I call a bottom side. We're not going to call it the bottom side. It is the basal side. It's the base. And we're not going to call it the top side. What do we call this region? The surface side. What do we call that? The apical side. These two are only true for epithelial tissue. Epithelial tissue have apical and basal sides. The rest of the tissues do not. They are connected in the same fashion, however. To understand how tissues work and to be able to identify them, cell shape is going to be super important. The structures they have inside of them are going to be super important to help us identify them. The way they are organized is going to be important to help us identify them. Are the cells close together? Are they far apart? How are they connected? In some cases, some tissues have fibers. Epithelial tissue does not have the presence of fibers. Helps us identify them. Okay. These things will help us determine what the tissues are and what they do. This is where I wanted to include it. There's the desmosomes. Do you see my desmosomes? They're connecting the two cells. Desmosomes will also connect the basal side of the cell to this region. This is the basement membrane. The basement membrane is kind of the floor of the cell, and that is where the cell is anchored to. In particular, that is where the epithelial tissue is anchored to the tissues below it, to the tissues on the basal side. Desmosomes hold them to those epithelial tissues. Hold the epithelial tissues to the basement membranes. That's how I should have said it. And then underneath, this is a different connective tissue. Okay. There are 4 types of tissues that we will be discussing in this class. Okay. We will do this later, so we're not doing it in this chapter. We're not doing nervous tissue in this chapter. We're not covering muscle tissue in this chapter. We are covering epithelial tissues, the ones that lines the organs. And we are doing many connective tissues, not all. Not all. There are a lot of them. I think we're only doing 10 in lecture, 7 in lab. It might be less in lecture. I can't remember off the top of my head. Reading guide, folks. Reading guide. Epithelial tissue. This is the one that makes the surface, lines the surface of organs. This one, we have to recall that we have to be able to identify them by shape, the cell shape, and the layers of cells to help us identify these tissues. How are these cells arranged? The first cell shape is squamous. What shape is squamous, folks? Flat. It is squamous literally means flat along one side. So these cells are flat. Sometimes they don't look it. This cell is flat. That's the top view. So from the top view, it is like this. It doesn't look flat. But when you look at it from the side view, guess what? It's flat. So it's flat along one side. That's what squamous means. These cells are flat. These cells are designed for two major functions. Since they are thin, they allow for a lot of materials to move through them. So they are designed for diffusion. They're going to be found in areas where lots of diffusion is required. Does anybody know an area where lots of diffusion is required? Your lungs. That's why they're lining the air sacs in your lungs. Lots of diffusion there, folks. They're also going to be designed to protect from abrasion. So from getting roughed up. Your skin is an example. Your skin is designed to get a little bit beat up. Not a lot. But when you bump up against the table, protects it from that. Okay. All right. So this is the shape is flat. Because it's flat, it allows these two things. The other thing we have to be able to identify is the number of layers. And the words for layers that we use, one of them is simple. How many layers if it's simple, folks? One single layer of cells. What's the other word we use to describe when it's two or more? Stratified literally means that it's layered. It has multiple layers. Two or more layers of cells. So this is what we use to describe epithelial tissues. Epithelial tissues are the only tissues that are going to be classified as simple or stratified. No other tissue, muscle tissue will not be categorized like that. Connective tissue will not be categorized like that. Did I say nervous tissue already? Nervous tissue will not be categorized like that. Only epithelial tissues. Epithelial tissues are named after their shape. Okay? It's important. Shape and are they layered? This is an example of a simple squamous epithelial tissue. How many layers? One. What's the cell shape? Flat. And we're looking at it from the top view. And it's an epithelial tissue. This one is our example, many layers. Two or more layers. This is our stratified squamous epithelial tissue. Many layers. I told my lab this, one big warning. This one can confuse us. Down here at the base, it is not flat. It is usually like a little ball, like a sphere. It's actually cuboid down there. We describe epithelial tissues by the shapes they are on the apical side, at the surface. At the apical side, it is flat. Don't describe it by the shapes at the base. It can get confusing. Cuboidal, what shape are these? They're cubed. Are they perfect cubes? No. What does that mean? That means when you look at them from the top view, here's that cell. It's as wide as it is tall. So the wide and the height are about the same. When you look at it from the side view, guess what? The length is about the same as the height and the width. So all sides are about the same length. Therefore, it's considered cube. Most of these are going to be spheres, like a ball, but it's still called cuboid, cube-like shape, because it's as wide as it is tall, as it is long. Look at this one here. Here's one, here's one, here's one, here's one, here's one, here it is. Be careful here, up top. This is the basement membrane. So here, this is one cell tall, and the one above it is also one layer tall. So here's another, a separate one. The basement membrane is in between. It's going to take practice to be able to identify whether it's simple or stratified cuboidal, because you will have to do so here. Okay. In lab, you only have to be able to identify simple cuboidal epithelial tissue. In lecture, you will also have to be able to identify one that is stratified cuboidal epithelial tissue. I'm going to ask you, where do you find this at? Your best bet is you find both of them make up sweat glands. Both of them. Simple make up the sweat that cools you off. The sweat that cools you off. Stratified cuboidal epithelial makes the sweat that makes you musky. What does that mean? That's the one that makes your sweat stinky. The one in your armpits, private areas, right? So your best bet when I ask you, where do you find these? Just tell me sweat glands. So we don't confuse these, okay? Both of them make different types of sweat glands. Columnar. What shape is this? Columns that are tall on one side. This cell, here is the base. The cell is one tall one. There's one. Here's another one. Here's another one. These ones exist also as simple or stratified. Most of them are going to be simple columnar epithelial tissue. That means they're only going to be one cell tall. Where do you find simple columnar epithelial tissue, folks? What's a good example? In all of your gastrointestinal tract, in the jejunum, in the duodenum, the small intestines, the stomach, the large intestines. These cells are great at secreting stuff I failed to mention this one. These cells, cuboidal, let's do this one first. Cuboidal, their primary goal is to secrete stuff. That's why they mainly make glands. Glands secrete stuff. They make stuff that is expelled from the cell. Columnar also does secrete, but it does something else that's critical in the GI tract. What does your GI tract also do other than secrete? And absorbs. It absorbs the nutrients that are you just consumed. It secretes the enzymes that you need to break down those foods. These tissues do both. Okay. All right. Simple columnar, GI tract, stratified columnar. Interestingly enough, stratified columnar is not common. It is actually considered a rare tissue. Technically, it is kind of part of your GI tract. The only place that I can think of where you can find this at is the anus. End of the GI tract. Instead of being one layer thick, it's two. Because they arrange in different shapes and sizes, the basal side is shown in green. Therefore, this is the apical side, considered the surface. Here, this inner ring is the apical side, considered the surface. This is the apical side, consider the surface. This is the apical side. Only epithelial tissue has apical, which is like the surface side, and basal. Only epithelial tissue, folks. Important to recall. I need you to complete this chart. Where do we find? Sorry, let's do the first one. Simple squamous. How many layers of cells is simple squamous, folks? What is the type of cell described by squamous? Flat, thin. What is its function? Diffusion and some layer of protection against abrasion. Where is it found in the body? What is it? Lungs, the alveoli of the lungs, the air sacs. This is part of your reading guide. I want you to do the rest of them. There are 1, 2, 3, 4, 5, 6, 7, 8. 5 of them are you're going to be double dipping for lab. It's kind of nice. They're not completely separate. What's that? Yep. One less thing to do. Where are they found? Here they are. Okay. Here they are. Look, I couldn't remember this one. The salivary duct. The salivary gland's one tissue, the salivary duct's different tissue. That's glad it reminded me. To be able to help you identify them, be able to tell me where they can be found. Okay. That's what I expect you to be able to do. That's going to be a little bit of your reading. And what am I going to ask you on an exam? I can show you a picture and it's going to point to it. Oh, name this one, that tissue. Name that tissue. This one's the histology. This one's an illustration of the histology. I can use one or the other, but I promise you, I will use an image where it is very clear to identify. So right now, talk to your neighbors, make sure we agree what that tissue is. How many layers of cells does it have? A single one. So right off the bat, no, no. What's the shape of the cell? No. Tall cell. What is the function of a columnar cell? What are you going to tell me? Secretion and absorption. How did you figure this out? It's one layer tall column cell. We just talked about that. Look at this last question. Where would you find this at? Intestinal tract? Right. A lot of the GI tract. You will even find this in the gallbladder. I think that's in your lab manual. Okay. The bile duct as well. Which one's this one? It's cuboidal. Is it simple? No. Look at this is one layer. Here's the second. Oh, that's a bad color. Here's the second. Do you see how it's made of two layers of cuboid cells? Here's the first layer. Here's the second layer. I told my lab this. These form donuts. Cuboidal always forms donuts because they make ducts and tubules and glands. Okay. Sometimes they're perfect donuts, and I have included pictures where they're elongated donuts. But you still see the cells, that line, that duct, that opening. Okay. What do these cells do? Primarily secretion. Where can you find stratified cuboidal epithelial tissue? What are you going to tell me? Sweat glands, the answer for both stratified and simple cuboidal epithelial tissues, folks. Apocrine sweat gland is the stinky sweat gland. We'll learn about that when we do the skin. For the most part, glands, 95% of them are going to be made of cuboidal epithelial tissue. Some are going to be simple, some are going to be stratified. So what happens is as the surface, there is part of the epithelial tissue. And what the glands and ducts are going to do is they're going to invaginate so that the ones typically at the base, this is going to be a gland. So that's going to produce whatever is secreted. And the rest of it is just going to provide like a corridor, a way for those juices to get to the surface. So the juices that it produces will leak out and come to the surface. And glands comes in all sorts of shapes. Look at them. Some do this curvy thing. Some are branched. Some are multi-branched. But they all do similar things, some sort of secretion. Glands are primarily going to be either cuboidal, 95% of them, and the rest of them are columnar. Here on the left, we have epithelial tissue. On the right, connective tissue. I'm going to pause it. And I want, how is epithelial tissue different than connective tissue? What's one of them? Fibers, that's good. Which one has fibers? Connective tissue has fibers. Epithelial tissues do not. No fibers in epithelial tissue. How else are they different? So epithelial tissue have specialized structures along the cell, specialized, like we talked about, the villi, the microvilli, the cilia, etc., that are associated with them. How else are they different? What is it? Location. Where are epithelial tissues typically located? At the surface of organs. And that's the internal surface or the outer surface. So keep that in mind. Surface of organs. Let's write the inner surface or outer surface. How else are they different? Different functions, but that's true within the epithelial and connective tissues. What's that? Which one has fewer cells? Connective tissues is not as dense. And by that I mean the cells are more spread out. Epithelial tissues, they're like right on top of each other, right next to each other. Look at the cells. There's one here, there's one here, there's one here, there's one here, there, and then the next one's all over here. They're more spread out in connective tissues. How else are they different? Very good. Epithelial tissues I mentioned are the only ones that are simple or stratified. None of the other tissues are like that. What do you call this surface up here? I almost put apical. Let's try that again. Apical versus the basal side only exists in epithelial tissue. Connective tissue has ground substance. What in the world is that? Who can tell me what that is? Uh-huh. So the matrix, because I failed to tell my lab this, I forgot, is the space between the cells. That is the matrix. Everything between the cells. Ground substance, that one I told my lab. What is that? That white space. And normally when that tissue is alive, it has specialized fluids flowing in the connective tissues. They have a lot more interstitial tissues among them, okay? Here's one that's not super intuitive. Connective tissue between these two will be the only one that can be innervated. What does that word mean? Has nerves in it. It'll have nerve cells in it. And it's the only one that will be vascularized. What does that word mean? It has blood vessels. Epithelial tissue, it's weird to imagine, does not have nerves in it. Epithelial tissue does not have a direct blood flow in it. It is weird to imagine, but we've seen this before. When you nick your skin just a little bit, you don't feel it. No nerves. You don't bleed. Not vascularized, not innervated. If you cut deep enough, it hurts and you bleed. That means now you're in the connective tissue area. Okay? So epithelial tissue is not vascularized, it's not innervated. All right. Connective tissues. One key way to identify them is they have fibers and they have ground substance. It's the fluid that is flowing between the cells. Okay. Before I move on, this is the poster child of connective tissues. What type of connective tissue is that? Areolar connective tissue. And there's a reason why areolar connective tissue is the poster child. And that's because it has pretty much a representative of every cell that can be found in connective tissues. It also has the 3 basic fiber types. So let's start with the fiber types. The large fibers, this one drawn in yellow. In the histology, it's going to be the pink thick ones. And because of their properties, they look like they're in the background. They just look like they're blurred out in the background, but they're not. This is a fiber made of a protein. That protein is collagen. This is the largest of the 3 fibers. Collagen fiber is huge compared to the others. It is thick, they are long, they are strong, they are for support and structure. They're the ones that lift your skin. That's why everybody's trying to get collagen supplement to prevent skin from sagging as we age. It happens. The medium-sized ones are these blue fibers shown here. Over here, they're the dark purple. This is, believe it or not, the medium-sized fiber. It is a thinner one. It is a protein. It is made of elastin. They are elastic fibers. These are not for support and structure like collagen. These provide different types of support. They flex, stretch, but what do after they flex and stretch? They return to their original shape. That's important. So they do stretch, they do flex, but they also recoil to their original shape. That's important. That's what it means to be elastic. The smallest one is so small, here it is. You can barely see it in histology, but it's there. These are called reticular fibers. This is the smallest fiber type. It's so small, it's hard to visualize. And they act like a web matrix to hold cells in place. So they have like a web, a mesh to hold things in place. These are your three fiber types, folks. They're all made of collagen protein, elastic protein, or reticular protein. Three different types of proteins to provide shapes to the tissues. And then we have different cells. There's a few that I want you to be familiar with, okay? The main one is this one that runs along the fiber. This one makes fibers. It is the fibroblast. Blast means it makes. It's called what it makes. It is a fibroblast. It makes collagen. It'll make reticular. It'll make elastic fibers based on what it's told to do. Which one's this big yellow one? Adipocyte. Also called fat cell. Oh, I spelled it wrong. Let me try that again. It is adipocyte. That is the fat cells. Areolar connective tissue has traces of fat cells in them. All of these, this one, let me try that again with a different color. This one, this one, this one, this one, this one, are all different types of WBCs. What are WBCs? They're different types of white blood cells that make their ways into the tissues so they can provide immune defenses. Okay. Those are the three main cells I want you to be familiar with that are typically found in connective tissues. Connective tissues can be loose, like this one, areolar connective tissue. That means there's a lot of space between the cells and not a lot of fibers. Loose, loose, a lot of space, not a lot of fibers. So lots of space, the space in between the cells is high. In other words, the ground substance is high. And relatively, the numbers of fibers are low. There's still a lot of fibers, without a doubt. There's still a lot, but it's not dense in fibers. Areolar connective tissue, all over the place. It's found all over the place. This tissue typically makes the basement membrane of epithelial tissues. This is why your lab manual says it's found everywhere. Because it is. And so everywhere. Adipose connective tissue. Someone's going to tell me, but this looks dense. It's loose because of the rule that the fibers are relatively low. There are fibers in here. They are hard to see. There are very thin fibers in here. That's the thinnest, smallest fiber type. What fiber types is that, folks? Which one's the thinnest, smallest fiber type? Reticular. So it's present, but it's hard to see. And the reticular fiber, what it's doing is it's holding all of these fat cells in place. Don't call them fat cells. They are adipocytes. It's holding those in place. Why do we have body fat? What is its main function? Energy source. What's the other one? I'm sorry, what was that? It helps maintain heat. You're missing the important one. Very important one. Provides cushioning. Surrounds your eyeballs. So when they move, they don't get damaged. You're moving your eyes all the time. Surrounds your heart, your lungs. As they beat, they don't get damaged. It cushions them. It provides cushioning too on the palms of your hands, the bottoms of your feet, and what you're sitting on. Provides cushioning, okay? Its function. This one's also found all over the place. And how to ID it. These two are easy, I think. The one that's hard is dense regular, okay? This one's gonna get confused with skeletal muscles at times. Very few cells, those are fibroblasts. Those make the fibers. Collagen fibers, look at all the collagen fibers. They are in the same direction. They're running parallel. That's what makes them regular. The collagen fibers run in same direction. This is dense because it has a high collagen fiber density. From your lab manual, where do you find this? Who remembers? Tendons and ligaments. They run in the same direction because these are designed to withstand forces either in that direction or that direction. And that's what your tendons and ligaments do. They only need to resist forces along one axis. So the collagen fibers run in the same direction. That is different than dense, irregular connective tissue. Dense irregular is one in lecture, not in lab. Look at it. Are they parallel? Look at the fibers. These are moving like this. These are in that direction. These are in that direction. These are in that direction. They are not parallel. It has a high number of collagen fibers, high density. That's why it's dense. And the fibers are not parallel to each other. All sorts of directions. This is dense irregular. Folks, this one's designed to get pulled in that direction, that direction, that direction, that direction, that direction. That's why the fibers are making various different axes all throughout this tissue. Does anybody know what organ gets stretched in all directions? The largest organ in your body. This one is found in the skin because it gets pulled in all directions. It can get pulled up. You can push it in. You can pull it to the side. This one is going to be found in the dermis of the skin. And we'll talk about that here shortly. I'm not going to give them all to you. There's 1, 2, 3, 4, 5, 6. Be very careful with this one. This one is elastic connective tissue, not elastic cartilage connective tissue. There is a difference between the two. This one is elastic connective tissue. Okay, so same thing. I want you to tell me its function, the types of cell, extracellular matrix. Tell me what the ground substance is like. The types of fibers, where you can find it. There's only 6 connective tissues that I need you to be able to ID and tell me about. It's not too much. 10-minute break. Now let's come back at 9:20. The integumentary system. This is the first system that we're going to cover in this class. Okay. Remember, organ systems are made of multiple organs, and organs are made of multiple tissues. We just finished our tissue lecture. So the integumentary system is made of multiple organs. What is one organ that is found in the integumentary system, folks? And that is the cutaneous membrane. That is the skin. So this layer is one organ, multiple tissues. The subcutaneous layer, or the hypodermis, is another organ. 2 or more organs working together make the integumentary system. There's going to be a lot of accessory organs. We're not going to cover them all. We will have to know what some of the accessory organs of the integumentary system are, like the hair, the nails, the sweat glands, sebaceous glands, and some of the muscles. Which connective tissue is this? That one should be easy peasy. Areolar. This one is found in the skin. This one is also found in the skin. Those are not found in the skin. We're going to have to identify where in the skin can they be found? What is their function? Just so you know, one of the functions of areolar connective tissues is to supply the epithelial tissue above it with nutrients. Remember, so I'm going to put supplies epithelial tissue. Supplies epithelial tissue with nutrients. Remember, epithelial tissue is not vascularized. So that means this gets oxygen, nutrients, vitamins, minerals, water. It gets it from its blood supply. And it will have to send it to the epithelial tissue above it so that they can obtain those materials. And it sends it via diffusion. It does not interact with blood, with the epithelial tissue above it, okay? That's why it's so important to the skin. This is the tissue that's going to nourish your skin at the surface. It is the superficial layer of the dermis. We'll talk about that here in a bit. This is like one of my favorite pictures I've gotten to prepare a cadaver like this. It's gross. People think it's gross. I think it's so cool. So is the integument an organ? Yes, that is the skin. Yes, it's your largest organ. The integumentary system is not. The integument by itself is. So they are two different words, and people use them interchangeably when they shouldn't. So be careful there. By the end of this lecture, I expect you to tell me what the difference between thick skin and thin skin. I've put this question on as a free response. Tell me in your own words what the difference between thick skin and thin skin. And the answer I got, thick skin is thicker than thin skin. Swing and a miss. That's all I had to say. Okay, this one is cool because this is a femur here shown on the right. And it's showing the different dermal layers. The dermal layers are all dermis, dermis, dermis. You have the epidermis. Epi means at the surface. So that's the top. This is what you see at the surface. The dermis is right below it. So it's deep to the epidermis. These two are the integument. The hypodermis technically isn't part of the skin. It is part of the integumentary system, but it is not the skin. This is a separate organ, and it is below. That's what hypo means. Hypo means below. Those are the three dermal layers. Underneath that, you have fascia, which connects the skin to your muscles. That's the muscle right there. So you have the layers up to the muscles. The fascia, depending on where you're dissecting it, can have a ton of fat on it. And if you cut an area that has a lot of fat, it is really neat with how much fat can be stored right in the fascia underneath the skin. It's quite remarkable. Like I tell my students, just think of the chicharron. It's delicious. So gross. That's what it is though. Deep fried skin. And it's fascia. No. No. Fascia connects things to muscle. Fascia connects things. Serosa makes like the inner lining of cavities. Okay. What are some functions of the integumentary system? What is the system of the skin doing? Excellent. Thermoregulation. How does the skin help with thermoregulation? Skin system, thermoregulation. Sweat helps you cool off. What else does the integumentary system? Let's list them. There's like 8 things. Let's try to at least list 6. What is it? Maintain body fluids. Excellent. It helps maintain hydration so you don't dehydrate. What's another one? Protects from what? Protection from abrasion. The skin protects. Your muscles are actually very weak compared to the skin. It also protects from something else. What is it also? So don't just say protection. You tell me protects from pathogens, bacteria, fungi, worms. What was and UV light. What else? Produces vitamin D. This is so important. This helps regulate your calcium levels. I have a friend who was very pale skin. He's a Southern California boy and went to do his PhD in Scotland. He got sun 3 days out of the year. People with pale skin who are in the sun area do not do well moving to areas like that. He got sick within a month. What did the doctor prescribe him? A light that he had to stand in front of every day naked for 15 minutes before he took a shower. His skin wasn't getting enough sun, wasn't processing vitamin D. They still make fun of him. I heard another one. Eliminates waste products. It does. It secretes stuff. On that note, it also absorbs. It can absorb water. There's a reason you put lotion on your skin. It absorbs those materials. Okay. It'll secrete and absorb stuff. We're missing a very important one. I think I heard it over here on the right. Sensory. So important. It allows you to touch temperature, textures. So important. There are more. These are the main ones that I want you to think of when you think of the functions of the integumentary system. The very top layer, the epidermis, forgot, that's what we're going to show here. The epidermis is all made of stratified squamous epithelial tissue. So the epidermis, the top layer of the skin, is all epithelial tissue. And 99% of it is stratified squamous. So that means this layer that is being peeled off, the top layer, is stratified squamous epithelial tissue. And it always blows my mind with how easy we forget this. Like I mentioned, you can cut yourself, and so long as you don't get deep enough, you're not going to feel it. It's not going to bleed because this epithelial tissue is not vascularized and is not innervated. You have to cut yourself deep enough down to here to feel it, and there's your blood supply. That's when you'll start bleeding. So the top layer, the epidermis, is all epithelial tissue. Most of it is stratified squamous epithelial tissue. Okay. Multiple layers, and at the top, they're thin, flat cells that are full of keratin. This is the hard calcium material that makes skin rigid. It can withstand some abrasion. What else is made of keratin in your body? Nails and hair. Guess what, folks? Preview. Your nails and hair are all from the epidermis. Okay. And glands. Almost forgot about that one. Your sweat glands are part of your epidermis. Your sebaceous, your oil glands are part of the epidermis. The epidermis is made up of many layers, and they're called stratum. So all of this is the epidermis, and it goes all the way to the bottom. And it always makes this like this hilly surface when the epidermis ends at the basal side. Let's start with the thickest layer of the epidermis. So this is the thickest strata. That one there is your stratum corneum. That is the most outer layer of skin. That is made of a lot of dead cells. Right underneath it, you have this really thin white layer. You can kind of see it in here, just that white little layer. That is stratum lucidum. Stratum lucidum. Underneath that one, it's the darker one. Stratum granulosum. The second biggest layer is this one that supports, sorry, let me change the color here. The second biggest layer here is this one, and that supports a lot of the pigments of the skin. That is stratum. Almost forgot. Spinosum. And the very base, the last one, again, we gotta pick better colors here. I'm gonna pick the purple. The very base is stratum, and I say basal, but it is basale. Folks, I struggle remembering the order of these. The good thing is it's gonna be multiple choice. And here's your mnemonic. Do you know what a mnemonic is? All right. Don't do this. Come. Let's get sun burned. Don't do that to your skin. That's the first one. The second one. The third one. The fourth one. I said I forgot that one. I was literally going, come, let's get sunburn in my head. And the last one. I need you to remember the order of these in the epidermis. So all of these make up the epidermis. They have different functions. That's too much for an intro anatomy course. But they do have different functions. One does a lot of protection. One grows new cells. One has a lot of pigments. And it is important eventually to know what they do. Not in this class. Just know the order. Come, let's get sunburn. Producing new cells. No, lucidum is actually to support corneum, okay? And this is where it gets interesting, all right? So, I told you earlier, I need you to tell me what the difference between thick skin and thin skin is. Thick skin has all 5. Thin skin only has 4 layers. Thin skin is missing, oh, wrong one, is missing lucidum. That one is absent in thin skin, present in thick skin. So they have different strata layers. That's the next slide, right? Thick skin versus thin skin. Thick skin has all five. Come, let's get sunburnt. Thin skin, come, get sunburned. No lucidum. No stratum lucidum in thin skin. Your body is like 90% or more thin skin. Where do you find thick skin? Does anybody know? Where's your skin the thickest? Your hands, the palms, your base of your soles of your feet, parts of the elbow, parts of the knee, and that's about it. Everything else is thin skin. Thick skin shouldn't grow hair. So hair can only be derived from thin skin because of the function. This one produces a lot of stratum, corneum, because it's constantly getting fluffed off. When you touch things, when you mess with things, when you walk barefoot, thick skin is designed to keep the corneum constantly producing so it can resist abrasion.
So is thick skin only skin that can also produce callus?
Some areas of thin skin can, but it produces it very well. Exactly. Okay, real quick before I move on, because we will. Please pay attention to at the end right here. This is the border between the epidermis and the dermis. And look at how it forms a mountain layer, even over here. That is super important. We're going to talk about that here shortly. And this exists in both thin skin and thick skin. So important. We'll get to that here shortly. So I expect you to be able to do this. I'm going to pause this for a minute. Put them in order from the most superficial to deep. Okay, which one of these? How many of these are part of the epidermis, folks? 5. 1 through 5 are all the epidermis. So the I did it again. The epidermis is making 1 through 5. So what is the most superficial layer listed here? Come. Let's get sun burn. Do you see how I did that? Come, let's get sun burn. Those are all part of the epidermis. The dermis has 2 layers, the reticular dermis and papillary dermis. I didn't give them to you on purpose. They're technically on the first slide. Which layer comes right underneath the dermis? Papillary dermis, then reticular dermis. Papillary, if you're not familiar with, means finger-like. This is a weird term. So that means whoever saw the papillary dermis, to them it looked like fingers. What in the world are you talking about, Professor C? This is a finger. This is a finger. Those are the fingers. That's why I pointed them out to you. So these regions are the papillary dermis. This is the reticular dermis. They have different functions, and we'll talk about them shortly. And then the last one, just by its name, is the hypodermis. Oh, sorry, I should have done blue. Hypodermis. And remember, this is also called the subcutaneous layer. Everything else is the cutaneous layer. Need you to visualize that in order. Your epidermis, the top layer, will replace itself every 45 to 90 days. That means from the bottom, from stratum bacilli, it's going to grow up in 90 days. It's going to fall off. And 90% of the dust in your house is just you shedding dead skin cells. Gross. So if the epidermis is constantly shedding, why are tattoos permanent? They go into the connective tissue. That ink is embedded in the reticular dermis, and that is why it is permanent. It is embedded and it stays there. The ink stays permanent. Its shape changes a little bit because as we age, the collagen fibers shift a little bit, and therefore it does some shifting, okay? Think about this. This is how I hope we're thinking about this. We should know right now what tissue is most of the epidermis made of. Which type? Stratified squamous. Not it. We talked about that one. Epidermis is made of complete epithelial tissue. This is not epithelial tissue. You should scratch that one off to begin with. Hypodermis, I didn't tell you this, but I mentioned this. This is the chicharron. This is where all the fat, the deliciousness is stored in the chicharron, the skin. So, are there adipocytes here? Do you see adipocytes here? Say it again. It's the retic. Why is it not the papillary dermis? Because let's go all the way to the beginning. I mentioned right here the areolar connective tissue is the region of the skin that is feeding the epidermis, the epithelial tissue at the surface. The papillary dermis is made of areolar connective tissue because the papillary dermis, its role is to make the finger-like projections. Do you see them right here? We saw them there. We saw them here. We saw them here, those finger-like projections. Why? It's giving everything above it the nutrients it needs because all of this has no blood flow. So the papillary dermis is made of areolar connective tissue. No, that is areolar connective tissue. The reticular dermis is the dermis layer that is designed to be pooled. It's keeping the skin structured. This is the one we have to blame for wrinkles. Oh boy. The collagen, once it starts losing its integrity, is when the skin starts to sag and wrinkle. Okay.
Does fiberglass start with more collagen or something? Or
The collagen just loses support over time. Your body doesn't make the proteins like it used to anymore. It stops making it. It doesn't make it the same. Just over time, it wears. Okay. All right. This is the area where the tattoo is embedded in. It'll stay within the collagen. It will still be the tissues will still be the same. But just like everything, over time, it does lose its integrity. Okay, it'll be made of the same stuff, it just won't be organized the same. Okay. So warning for you guys, I mentioned this to my lab. People like to put collagen on their skin. Do we know what collagen, what type of nutrient, what type of molecule is collagen? Do you think your skin absorbs proteins right through it? No, it does not. How does your body get collagen? By what you eat, not what you put on it. It's $1 million company, business. I'm not convinced it works. You're just, anywho,
is it lotion? What is it?
it's in lotion, right? Because everyone hears this word collagen, they just want to put it on everything. You're getting what you pay for, but is it helping you? I don't know. If you do take collagen supplements, we know where collagen comes from, right? It's not vegan. Where does collagen come from? Like cow joints and cow bones. So that white powder stuff you put in your coffee, it's ground up bone. Don't give it to your vegetarian. Don't give it to your vegan friends. Okay. If you do look at it up and you do find vegan collagen, it's expensive, but it's all made in a lab. Just so you know. Okay. Yes, collagen that you consume can help your skin, not what you put on your face. Okay. How does this structure help the function of the skin? It helps the skin maintain its integrity, its support from being pulled in directions, right? That's why it's dense, irregular, connective tissue. This one's important. I'm going to age myself. Nelson. Who knows who Nelson is? The guy in the corner. Thank God. For a while, it was getting fewer and fewer students. And I was like, oh, man. I remember watching this episode when it first came out. But there's an episode, summer's over, but summer started, and Bart gets a swimming pool, and he's going to jump off a tree. And Nelson, the jerk that he is, tells Bart, hey, your epidermis is showing. Bart freaks out, falls, breaks his leg, summer's ruined, no more swimming pool. Is Nelson right? Absolutely. Your skin is showing. Your hair is showing. Your nails are showing. Your glands aren't showing, but they are part of the epidermis. That is the point of the slide. All of those things are your epidermis. So, of these three, which one produces sebum? Easy peasy. Does everybody know what sebum is? Your skin slash hair oils. They keep your skin hydrated. They are produced by the sebaceous gland, which are made of dead cells filled with hard keratin. Your nails. Just your nails? So is your hair. Your hair and your nails are made from the epidermis, and they are just dead cells that are filled with hard keratin. Made of the same thing. Oh, I forgot one. What do you have in high abundance in your palms, your plantar, and your frontal region? I forgot to include it, so I'm gonna write it in. And guess what? Your sweat glands also come from the epidermis. They're part of the epidermis. Okay, now this question, which one is produced from shafts from follicles? Hair, that one is specific to hair. Hair forms shafts, nails form beds. And guess what? Your hair, your nails, your sebaceous glands, your sweat glands, both type of glands, sweat glands, are modified versions of your epidermis. That is very important, folks. These are all appendages of the epidermis. So this is how I've asked questions on the past when it comes to the skin. Your epidermis is made of which tissue type? Epithelial tissue. The hypodermis is going to be predominantly what? Adipose. Your papillary dermis, areolar. Your apocrine sweat glands, what is it going to be made of? Epithelial tissue. Your hair follicle, epithelial tissue. Your nails, epithelial tissue. What part of your skin is made of dense, regular connective tissue? Yeah, nothing. Not regular, irregular in the skin. The hair is an appendage of the skin. And to be exact, the hair is an appendage of the epidermis. So here we have the epidermis. This is the top layer. And look what the epidermis does at the skin. It'll invaginate and it'll form a crevice and then it'll come back up. The glands, and then it comes back up to become the epidermis. So the hair and the glands are part of the epidermis. And the reason I'm showing you this slide is because, yes, it does go down. The hair is at the same level as the hypodermis. Look, there's all the fat. That's the hypodermis. Oh, hypodermis. There's all the fat. But you see that it's still coming off of the epidermis because the epidermis has curved and gone down deep. Still the epidermis, folks. Still part of the epidermis. So students see this and they're like, look at how far down the hair goes. It's part of the hypodermis. No, it is a modified version of the epidermis. Okay. Hair is only found in thin skin. I'm going to tell you a story that I probably shouldn't tell you, but I'm going to tell it anyways. Hair grows from the bulb. That region is the hair bulb. Hair grows from there and it grows up. Oh, let me pick a better color. And it grows this way. From down here. And it'll grow up. Why do I mention this? Well, I'm not going to say my friends. This is my friend, and I'm serious when I say this happened to my friend. Shortly after I graduated from my bachelor's, my friend got a call. We're going to offer you this job, but you have to come take a drug test. We're going to take a sample of your hair. And my friend thought he was so clever when he showed up to work after cutting his hair, buzzing it completely. And they're like, all right, whatever. He worked for a couple of weeks. They didn't say anything. His hair grew out and they took that sample and he didn't have a job. Why? Well, when he was participating, this part of the hair exhibited what he participated in. And it grew out to here. My friend is not very bright. He thought hair grows like it just, like that's all new hair. Like it grows at the edge. My response to him is they should have fired you just from that fact. That's how you thought hair grew. And many people think that. People think that the hair grows from the outer edge. No, it does not. A lot of people realize this when they start seeing the color come in, right? They say, oh, look at my roots, because of how hair grows. People don't recognize that, and it blows my mind to this day. Okay. Hair is created by the follicles and the bulb. So the follicle is this protective layer in blue. This is your hair follicle. This holds the epidermis that makes your hair. Here's the epidermis that makes your hair. This is also part of the follicle. Both of them. You have the outer follicle and the epidermis, that's the follicle. Those will provide the nutrients and support for your hair. Everything in here is different layers, different types. Is it keratin? I'm brain farting right now, how to spell keratin. That's the hair shaft in there. It's made of different cells of keratin. In the very center, you have your soft keratin. And then the outer layers, this is all made of hard keratin. Keratin is a calcium protein that will make hair. Okay. And it grows. Do you see this little dark thing? That's where all the hair grows from. In the hair bulb, you have this structure called the hair matrix. This is the only part of the hair bulb that makes new hair. So what happens is as we age, the hair matrix dies. What happens to that person? Balding. No hair matrix, no new hair. That's the part that kind of goes away. Okay. All right. So in this image, in black, there it is. This is the hair matrix. That's important. This is also important. This part right here that I'm outlining in red is not part of the epidermis, but it incorporates itself with the hair right above it. That part that I circled in red, let me make it a little bit bigger. This is technically a modified version of the papillary dermis. Therefore, its name is the, let me try that again, trying to jump the gun, is the dermal papillae. Papillary dermis, but it's the part of the hair. The dermal papillae is responsible for providing the nutrients from the blood that are required for the hair matrix to grow new hair. Both of these are part of the hair bulb. The hair bulb is kind of the base of the hair shaft. The hair shaft is all of it, all of the actual hair, no matter how deep it is. That is the hair shaft. The follicle is the purple and blue stuff that surrounds the hair that provides the nutrients and maintenance of the hair. Has anyone ever had like a kid pull their hair out and it comes with all that white stuff? That white stuff is the hair follicle. That hurts. That hurts. All of that pain is coming from the dermal papillae because in addition to having the blood supply, that's where the nerve supply is. Ouch. Sweat glands are also made of epithelial tissue. You have your apocrine sweat glands. Apocrine are the larger sweat glands, and these are the ones that make your stinky sweat. Eccrine, oh, did I do that wrong? Eccrine sweat glands, no, I did that right. Eccrine sweat glands are the ones that make the fluid, the sweat to cool you off. So that sweat has different functions. Apocrine is designed to have a scent. So people around you kind of know what's happening to your body, pheromones. Eccrine is designed to simply cool you off when it's too hot. Okay. Apocrine is made of what is it? Brain fart. Stratified cuboidal. Cuboidal epithelial cuboid. I can't think. It's getting to that time, guys. Stratified cuboidal epithelial tissue. I apologize for the typo. Eccrine is simple cuboidal epithelial tissue. So when I told you, when I ask you, where do you find stratified cuboidal epithelial tissue? Sweat gland. Where do you find simple cuboidal epithelial tissue? Sweat gland. It's your sure bet. I failed to mention this, so let's mention it now. The hypodermis, do you see that? That is all adipose connective tissue. When you start getting deep enough to the skin, where it's mainly made of adipose connective tissue, you are in the hypodermis. I failed to mention that earlier, I apologize. Keep in mind, this is, let me pick a better color. This is still the epidermis, even though it's pretty much all the way down to the fat. It's still epidermis because of how it curves down into the skin. This one's my favorite. Don't be shy. Show people your luneals and your epiniciums. Your cuticles, your nail edges, your luneal. That's your luneal. Epinicium. It's like where it comes in contact. Some people call that the cuticle, epinicium. This one also has a hair matrix, a nail matrix. That is where the nail is formed. Has anybody ever lost a nail? No, remember how it takes a while and then it grows out this way? Takes a good while to regrow a nail. All of it that comes in contact right here, all of that is the nail bed. And the little white thing right here, where it comes off, this is called the free edge of the nail. Does anybody know what nails are for? Don't tell me they're for fighting, because we don't have to fight or kill our prey with our nails. I didn't realize how much nails are important until I lost my thumbnail and one of my middle fingernails. I shouldn't be allowed to use power tools. That's what happened. But what are your nails for? Does anybody know? It helps your fingers maintain their structure. They act like a backboard for your nail. When I lost the one on my thumb, it was really hard for me to hold a big cup because I didn't have the support on the back of my finger. That's like the main thing they do, is they provide support for the tips of your fingers for when you're manipulating things, messing with tools, grabbing a cup. I didn't realize it until my nail was gone. It's a weird feeling. This is our last slide of the day. We made it, folks. We'll talk about this, right? The hair matrix. This is the part that makes new hair. What does the dermal papillae do? It provides the nutrients to the hair matrix so it can make new hair. What is the hair shaft? the actual hair. Okay, just the actual hair. So which one of these would you need to transplant so that your patient will grow new hair and keep hair? What's the answer? D. You need the part that makes the new hair and to be able to nourish that part that makes the hair. That's what they do. When they pull it out, I've been told this is extremely painful. They only need to pull out, technically, just this part. So only thing that needs to come out of the hair and be put into the patient so they can grow the new hair. Here in red, this is the dermal papillae. I'm doing it in red because that's where the blood flow is. That's where the nutrients are supplied. And look at these very few cells. That is the hair matrix right there. That is all that is needed to really allow the person to grow new hair. The hair matrix are like germ cells, they're stem cells that make hair. And for funsies, look at this. These ones that are brown, those are your melanocytes. What are melanocytes? They produce melanin. They produce pigment in the skin. They make my skin brown. They also produce skin pigment for the hair. What happens when the melanocytes die? What happens to the hair? It turns white, like all the ones on my chin. It was a sad day when they started to go. Right? It's my hair matrix is still working, the dermal papillae is still working, but the melanocytes are done. Hair comes out white without pigment. Okay? All right, this is where we're wrapping up for the day. Please submit your cards on your way out.